Okay, this series looks at Mycobacterium, Streptococcus, and Neisseria genera. These are three different genera of bacteria that do contain human pathogens. This correlates to exercise 38, 41, 42, and 43 in the lab manual. Exercise 38 deals with Mycobacterium. This includes species like Mycobacterium tuberculi, Mycobacterium leprae. You've already been exposed to those when we talked about the acid-fast stains. This particular genus of bacteria tends to grow very slowly, so they often need an extra enrichment medium to assist them. Common one that we use is called Lowenstein-Jensen medium. Usually this will be in test tubes, not on a plate. In the test tube, the media is a very kind of light bluish green, almost like a turquoise color. And when you grow, depending on the species of the Mycobacterium, when you grow it, it'll be a whitish, off-white, kind of yellowish, crusty color to it. It does grow very slowly. Unfortunately, I do not have an example of this on the video link just because of the slow growth of it. But just to give you an idea, it often does need this enrichment medium. Exercise 41 deals with culturing Streptococcus, commonly found in the mouth. In the upper respiratory tract, some species may commonly be found there that are non-pathogenic. Other species can be pathogenic and cause strep throat. So in exercise 41, there is a video linked to that does show a lab demo of doing a throat swab and then inoculating a blood plate. A blood agar plate is considered an enrichment medium to help assist the growth of the Streptococcus species if it is present there. One thing about Streptococcus, it could grow at regular lab conditions in an incubator. However, the colonies would typically be very, very tiny because it prefers a lower oxygen concentration than that which is found in normal atmospheric air. So to assist or try to enhance the growth, you would incubate the medium but put it into an anaerobic container, or in our lab we use a candle jar, and that is also seen on the video link. Essentially, you would put the plate in a jar, we light a candle, close the jar up. When the candle burns out, that means the oxygen level is lower. And so it just helps to assist the Streptococcus to grow better. After about 24 hours of growth, you would look at the culture, and you thing that you're looking at because you inoculated a blood plate is to look to see whether the red blood cells have been broken down or hemolysis has occurred. There's three main types of hemolysis that could occur. It depends on the species. There's alpha, beta, and gamma. Gamma hemolysis is when there is no hemolysis, so you don't see any change in color on the blood agar plate at all. Still looks red, and the bacteria growing just looks like your normal kind of whitish, off-white, maybe yellowish color colonies growing. But there's no change in the red coloration of the agar. Alpha hemolysis is when near where the colonies are, or near where the streak is, not the entire plate, just near where there is growth, there may be a little bit of a greenish hue or greenish brown coloration near the colonies. That would indicate alpha hemolysis. Beta hemolysis is when you have complete clearing or complete hemolysis of those red blood cells near where you have growth. So where there is no growth, it may still remain red, but around the growth, there's complete clearing, as in you could put a newspaper underneath the petri dish and you could read it. This is what you would be most concerned. Those species of Streptococcus that cause beta hemolysis are your most pathogenic ones. So if you saw that on a plate, definitely don't open the lid. Keep it closed. Exercise 42 also dealt with Streptococcus. This was dealing with looking at or trying to isolate streptococcal colonies, organisms that are in the oral cavity in your mouth, specifically along the gum line where the gum meets the teeth. They're often found there. So a swab was done, and then two different types of media were inoculated. Mitis salivarius agar, which is a petri dish. This is a selective media. It does contain dyes and certain chemicals that will inhibit the growth of most other organisms so that only Streptococcus will grow. And then Todd Hewitt broth, this is a test tube with broth in it. That is an enrichment medium also to help assist or enhance the growth of the Streptococcus species that they are present. Both the Mitis salivarius and the Todd Hewitt mediums were also inoculated in a candle jar and observed after 24 hours for growth. Exercise 43 deals with Neisseria. Neisseria often grows best if it has an enrichment medium, so sometimes it's grown on regular blood agar or on what we call chocolate agar, or sometimes referred to as chocolate blood agar. It does not contain chocolate in it. What happens in the preparation for the chocolate agar is you take a regular agar, like a nutrient agar, tryptocase soy agar, and you mix red blood cells with it. In the process, the red blood cells burst and hemoglobin is mixed in with the media. So when you end up pouring the plate and making that final medium, when you look at it afterwards, it has a very dark color. So it kind of looks like chocolate, but it's not. The purpose of that, once again, is just to help enhance or assist, hopefully, to get good growth of the Neisseria species. Some people will say that you should grow Neisseria in a candle jar, but you don't have to. You can if you want, but it should be able to grow under normal atmospheric conditions of oxygen levels, so it would be fine.